Okay, I appreciate this opportunity to talk to you today about something I have found to be extremely interesting. This study was actually predicated on a very simple question from a natural resources manager who asked me about how does hickory regenerate. He didn't ask how we regenerate hickory, he just asked about how hickory regenerated. And I asked some of my colleagues, and I've looked into some of the literature, and I found out that we really know very little about hickory regeneration. We just haven't put much effort into any research on that. And part of that reason probably is because we have not valued hickory as a timber species. It's one of those secondary species and has low value in the past. And it also didn't have high value as a wildlife species. We certainly recognize it as playing a role in the biodiversity of our forests, but there wasn't really any impetus to be in researching hickory. So I had the opportunity to take some data from a large-scale study that I had that looked at hickory regeneration in particular, but I'm also going to talk about regeneration of some other species as well. And what also makes this interesting for me is we work in these upland hardwood systems, and we call them oak hickory forests, and yet we haven't spent any time really doing any research on the hickory part of these forests, but just the oak part. So I will talk a little bit about the oak today too. So we're going to talk about how hickory regenerated under 5 silvicultural prescriptions in an oak hickory forest, and this research is actually taking place in northern Alabama on the Cumberland Plateau. Sorry about that one. This is just a graphic outlining the Cumberland Plateau, and you can see that it's part of several states, the majority of it being in Alabama, Tennessee, and Kentucky. It is a true plateau in which the plates pushed against each other, and the area that became the plateau actually rose straight up and then over time has eroded. And we do have parallel or striated rock, and it is a true plateau, but we have a lot of topographic relief because of the erosion that we've had over time. And this is just a slide depicting the stands that we're looking at for this study, and they are located in the northeastern part of the state of Alabama, again on the Cumberland Plateau, and these are typical upland hardwood stands for this area. So a little background on northeastern Alabama. I just wanted to, for those of you who haven't been there or may not know what it looks like, one of these pictures is northeastern Alabama, and it happens to be the picture on the right. We are the interior uplands in the mid-Cumberland Plateau region. As you can see, as I mentioned, we have a lot of topographic relief because we've had a lot of erosion over time. Our forests are mixed upland hardwoods, and of course this does include a pretty strong hickory component. And we have a lot of management uncertainties. We have the same problems plague us here as in the southern Appalachians and in the Ouachitas, in terms of trying to regenerate these stands, especially on better sites. And some of our management uncertainties just come from the lack of knowledge that we have about how we got these forests that we have today. We can guess about some of the disturbance regimes that gave us these forests today, but we really don't know what precipitated the forests that we have. We know that, for example, American chestnut was a large component of these forests and of course died out over time. There was probably a lot of selective logging in these stands over time. There was probably other disturbances such as grazing, certainly weather disturbances, and so forth. And so there's been a little bit of research looking at management, but really we know very little about hickory. As I said, who studies hickory regeneration? And the answer to that is very few people. So why are we interested just in the Cumberland Plateau region in general? A lot of you may not be real familiar with the Cumberland Plateau. This quote came from Gene McGee. He was a silviculturist with the U.S. Forest Service located in Suwanee, Tennessee. And this quote was from a good number of years ago, 1978, in Southern Lumberman. And Dr. McGee said, "Nowhere in the South is the opportunity greater for increasing forest benefits than on the Cumberland Plateau of Alabama, Tennessee, and Kentucky. This area of millions of acres of commercial forest land is a sleeping giant." So you can see Dr. McGee saw a lot of potential in this region and did a lot of research to capture and have some knowledge on that potential. And we're going to contrast this to a more recent quote. This was from June of 2000, and a really different venue, the Washington Post. And this author said, "From an airplane at 1,000 feet above, the steep hill along the Cumberland Plateau looks as it has been scalped, a muddy, trampled contrast to the surrounding green. Nearly every tree on its slopes has been shorn, then fed into a giant chipper nearby." And the quote ends with, "This is not your grandpa with the crosscut saw anymore." Those of us that are forest managers and are very concerned about the natural resources in this area have a lot of heartburn over this type of press, and it's unfortunate that this is out there. So one of the reasons that we are interested is that we need to be a little more vocal and have a little more knowledge so that this type of press isn't as prevalent as it happens to be today. So why are we interested? Well, what we want to know is, how did millions of oaks and hickories on good sites become established between 1820 and 1920? What were the conditions that allowed today's stands of oak, ash, hickory, and poplar to become regenerated? And I mentioned we had a lot of different disturbances, but we don't really know the history of those disturbances. We don't know the intensity, the duration, and those types of things. We have a lot of studies that have shown that site quality influences regeneration difficulty, and I'm sure others in this short course have spoken about that. We know that lower quality sites are easy to regenerate, especially to oak. Not so much is known about regenerating it to hickory. And we know that higher quality sites are more difficult to regenerate, again, in particular to oak. So what does this have to do with hickory? Well, we know that different factors can limit oak regeneration, and we know some treatments that can control some of those limiting factors. For example, we know that light is often the most limiting factor in a lot of our systems when we're trying to regenerate the species, select species. And we can control light by altering different levels of the basal area. And we know that different levels of light allow oak to respond differently. But what we don't know is how hickory would respond to those same different light levels. We also know some things about the probabilities of success of different types of regeneration on different sites. So, for example, we know that small advanced oak reproduction is probably not going to outcompete a stump sprout of yellow poplar, for example. So we know seedlings might not be as competitive as seedling sprouts, which might not be as competitive as stump sprouts. But we really don't know about these probabilities for hickory. We can use the knowledge from some other species, but we don't have these probabilities for hickory in particular. So lastly, another reason that we're interested, I took this picture a couple years ago at one of the local large box stores, and these are hickory cabinets. And the story goes that in the early 2000s, there was a home show in Atlanta. And at that home show, they showcased, in particular, hickory cabinets, but also hickory flooring. And with the changes in technology and the changes in people's aesthetics and what they want in their kitchens, this hickory cabinet these cabinets are very appealing aesthetically, as you can see in this picture. And as a consequence of that, the actual price of hickory has gone up to about $2,000 per 1,000 board foot, and it has, or $200, excuse me, per 1,000 board foot, and it has stayed there for the last couple years. This is data that was provided to me by one of the logging yards in northern Alabama. And when I asked him about, this hickory, and it used to be you couldn't pay a logger to move hickory, and because of that, what we've ended up with is a lot of high quality hickory and even veneer quality hickory. And the quote from the timber manager of this company said, "We searched and we researched these stands, and all that was left was hard maple and hickory." In other words, they've kind of selectively harvested a lot of the other species out of here, which actually gave an edge to allowing hickory in particular to continue to grow. It's a little larger. It's taken up that growing space when those other species were removed from there. And as I said, we're growing some veneer grade hickory that now is a little more economically viable to take out of the woods. So just to review some of what we know about oak and a big theme, David Loftus spoke to you earlier and has spent his career looking at regenerating oak stands, especially in the southern Appalachians. And his seminal paper said that oaks demonstrate better regeneration on lower quality xeric sites. And for us, this would be the top of the plateau, the flat tabletop that's slightly unundating. The site index, which for us would be the height of a tree at the base age of 50. For upland oaks on the top of the plateau is 65. So we probably don't have as many concerns with regenerating oak on the top of the plateau versus the side. David said that oaks are poor competitors with other species, particularly yellow poplar and other shade-tolerant species on good sites. For us would be the plateau side or the escarpment versus the tabletop. And the site index here for upland oaks is about 80. And then on the bottom there I have, well, what about hickory? Well, we have no idea what the site index values would be for hickory. All we can do is relate those to their cohort there with oak, but we don't have these same values for hickory. So the objective of this study was to quantify the sources and species composition of forest regeneration resulting from a range of site disturbances, including looking at how hickory was going to respond. And a goal is to help forest managers understand how the stand manipulations will influence current stand dynamics and the future composition and structure of these stands. And finally, the landowner on which this land was done. The landowner wanted a positive financial return from a timber harvest, and he wanted the sustainability of the oak component. But the more that we worked with this landowner, the more we realized that he certainly didn't want a monoculture of oak. What he wanted to do was he really wanted to maintain the mixture of species that he had out there. And I made the comment that he wanted to hedge his bets towards maintaining the biodiversity, and that certainly would include the hickory in these stands. So as I mentioned, the study area is in the kind of northeastern part of Alabama on the mid-Cumberland Plateau. And this is just a circled area about where it is in our physiographic regions. And it is on the escarpment or the side. And these are fairly good sites. They have deep to very deep soils. They're well drained with moderate fertility, and they have mostly loamy soils to gravelly loamy soils. The slope ranges from 15 to 20%, although some people have argued that it's a little higher in places when they come out and do some work with us, but it's around there. The site index, again, site index being the height at age 50, for upland oaks is 75 to 80. We have no idea what it is for hickory. The site index for yellow poplar is 100, and the oak reproduction is low in number and small in size. Again, we've done a lot of research on oak, so we know a lot more about the dynamics of the oak, but the hickory also is very small, where we have advanced reproduction, and it's also not very many seedlings are out there. So what we wanted to do was, I mentioned earlier that we know that controlling the light will influence how these species will respond and then regenerate, and then how competitive they will be to be a component of that next stand. So what we wanted to do was alter the light of these stands incrementally. So we did this by retaining or removing the basal area in 25% increments. So we'll start at the top. We left 0% residual basal area, or we clear cut the whole thing. So we didn't retain any. We had a residual basal area retention of 25% of the total basal area, which meant that was a commercial harvest where we took off 75%. We had a residual basal area of 50%, again, another commercial harvest. And then we had a residual basal area of 75%. As you can imagine, this was not done commercially. This was done using an herbicide that we used a hack and squirt method to reduce the mid-story. David Loftus has done this in the southern Appalachians, and I'm referring to it as a Loftus oak shelterwood method because we used his method exactly and tried to reproduce that over in the Cumberland Plateau, and I'll talk a little more about how we went about that. And then finally we had a residual basal area of 100, or we had a control. Treatments 2, 3, and 4 are really true shelterwoods. The overwood will be removed in 2 steps. And the second removal, we hope, will come 10 years or so after the initial. Again, our objective here was to use the basal area to change the amount of light that's reaching the forest floor in the reproduction, and seeing how that light will influence the regeneration in order to grow some of the small regeneration into a more competitive position but we don't want to let so much light in that the competitors we don't want take off. So it was a true experimental design. This is not a case study. The treatment units are 10 acres in size. At the time, that was the largest clear cut that this landowner wanted to make. Each treatment was replicated 3 times in a randomized complete block design. We used analysis of variance to quantify the treatment differences, and we used a range test called Duncan's multiple range test for means comparison. And I'm not going to get into the statistics any more than that, but I just want you to know that these are statistically valid inferences. These aren't just a case study in which we've made some general observations. So again, the study took place in the north part of Jackson County, and the little blobs on there, the purple ones on the right are 2 replications, and the light blue ones on the left are another replication. And it's just always interesting to look at the topo map, and you can see we are on the escarpment or the side versus where the word plateau is written. You can see the lines are really far apart, and that's the top of the plateau, which would be much more drier sites than the side. And this is what the layout looks in the field. The different colors are just depicting the different treatments, and they were randomly assigned to these 3 blocks. 2 of them are at Jack Gap, and 1 of them is at Miller Mountain. And again, they were 10 acres in size. To take our measurements, we put in some permanent measurement plots, and there are 5 of these in each of the treatment units. And these are 3 circular concentric plots that are in each measurement plot. And then you can see a couple green dots on there. We added some additional regeneration plots as well. So our measurement plots are, the smallest one is, we are taking the regeneration measurements on, and this is a 100th acre. And it looks like anything that's on this 100th acre, we actually enumerate all the species by species and height class, and then we tag a sample number of seedlings, and that's what you're seeing here. You can see this is an oak seedling, and this is a brass tag with a 60-penny nail that we've put next to it. And even if this gets damaged during the harvesting activities, we usually can find this, and if it sprouts or if it's completely removed, we're able to find the seedlings. And this is a great way for us to follow these populations over time. Then we have a slightly larger plot. You can see depicted by the number 2 here. And this is a 40th acre plot in which we are looking at all mid and understory measurements, and I'll get into those details. And then finally, we have 1 larger plot, which is the overstory plot, and this is a 5th acre plot in which we're looking at the trees that are commercially viable. And this is a picture of one of the small tags that we put on all the trees out there. The seedlings get the brass tags, and the trees get these aluminum tags. So for the seedlings, as I mentioned on this small plot, we look at species number and height class of every single seedling that's out there, and then we take a representative sample of 5 to 10 seedlings, and we map those. And so we measure the distance and azimuth to the permanent plot center, and then we tag those, and we remeasure these annually. For the 40th acre plot, everything that's 1.5 inches tall and greater, we measure species DBH, and we map and we tag these and remeasure these annually. And this plot really is to help us look at things, especially sprouting. So, what kind of sprouting probabilities can we expect from some of these smaller trees out there? And finally, we have the overstory, which is everything that's 5.6 inches taller and greater. And again, we've mapped and tagged all those species. We looked at canopy cover a couple different ways. We used hemispherical canopy photographs, which was the picture at the beginning of this presentation. And we also used a handheld densiometer to capture the amount of canopy cover we have. And then, as I've mentioned, the reason that we've set up these experiments is to alter the light levels to see if we can grow some of the small advanced reproduction that we want into a more competitive position. Under which light levels would that be most enhanced? And we're certainly adjusting basal area, but at the same time we're altering the light. So we have a device called a septometer, and it has a small computer and a wand that has photoreceptive cells on it, and we look at relative reduction in photosynthetically active radiation in these treatments compared to what you would have in the open. So this is kind of the nuts and bolts of the study. The trees were marked to stay. As I said, 3 of them were active harvests. The leaf trees were chosen on the basis of 3 things. We favored oaks, ash, and persimmon. Oak and ash for the timber value, persimmon because there's not much out there, and they do hunt on this site, and he just wanted to maintain all the persimmon he could. And we also chose the leaf trees based on crown class, and we worked with John Hodges on this a little bit, and he helped us with the marking. And we left dominant and co-dominant trees, and we also left very high vigor trees. And of course, we had to leave other species besides oaks and ash, but those were preferential leaf trees. The harvesting and the herbicide treatments were done in the fall of '01 and the winter of '02. The harvested trees were chainsawed, felled, and skidded along pre-designated trails. First, we daylighted the roads in the early fall, and then the harvesting proceeded through the fall and into the winter of '02. For the 75% retention, if you remember, this was the herbicide treatment. We used arsenal to target the mid-story species, and we did this with hack and squirt method, and we just had a small crew, and we did this ourselves because we had to be able to select the trees that needed to be herbicided and be able to identify those species. So an average about 381 stems per acre were treated this way, and they had a mean DBH of about 3 inches. And the goal of this treatment in particular was to not create any holes in the overstory canopy. And what I'm trying to show here is this is a fairly large tree. It actually was an intermediate crown class, and a little bit of a hole was created, but not a huge hole here. And this is just a picture of one of the trees that we hacked, getting through the cambium. Very, very small amount of herbicide used. We used arsenal. The active ingredient is imazapyr because it targeted all of the species that were in our mid-story that we wanted to target. And again, the main goal of this was to alter the light so that the very small advanced oak reproduction could grow into a more competitive position. But we didn't want to let so much light in, again, this is a very good site, that yellow poplar would take off. So we're trying to guess what those light levels would be, and this was a treatment that David had used that had done this very successfully. We actually had no idea how hickory would respond to this, so no information on it. So these are some pictures of what it looked like after it was implemented. And you can see there's the 50% versus the 25% retention. And then the control and the clear cut. And this is actually the control, and I'm standing in the 25% retention overlooking one of the clear cuts. And finally, the 75% retention. Is very difficult to get a photograph of this, but I'm hoping you can see that in this area here, we're lacking green vegetation, the same thing here. And you certainly can see patches of increased light getting on the forest floor. And to try to get you to see that mid-story being reduced, I tried to splice 2 photographs together. This is the untreated control with this tree being similar to on this plot, and then I just turned and took a picture over here of the treated area, and you can see there's a lot more mid-story growth here than there is in this 75% retention. So a gradient of treatments going from a control, missing the mid-story, the 75, the 50, the 25, and then of course the clear cut. So we did set up this gradient here. So how did we do? I'm going to show a few charts that look like this, and again I'll always refer to the treatments as the percent retained. So on the left-hand side, we go from 100% retained to 0, from a control to the clear cut, again the 75 being the herbicide treatment. The next column is the pre-treatment basal area, and that's in square feet per acre. And you can see they averaged a little over 100 square feet, which made this really nice to talk about, because if you take out half of 100, it comes down to about 50. And if you look at our post-treatment basal area, you can see these are the numbers that we got. Again, a nice gradient going from over 100 to 5 square feet. When people ask how we had 5 square feet of basal area in a clear cut 1 year post-treatment, it was a commercial clear cut, not a silviculture clear cut, and there were some small stems that were left out there, and of course we were able to pick up in our tallies. And if you look at the actual retained, which is in the last column, we had 1 tree that died in the control. That's why we don't have 100%. We were pretty pleased with that. And we had 5% that we actually retained in the clear cut. Our goal was 0, of course, so we were pretty pleased with that. For the herbicide treatment, our goal was 75% retained. We got 70. We're very pleased with that. For the 25% retention, we got 28% retained. We were also very pleased with that. But you can see the last one I'm going to talk about is the 50%. We ended up with 38% of the basal area actually retained. And we just happened to mark these stands a little heavier. Remember, there were 3 of them that were treated this way. We did use a prism while we were painting trees, and we did check. It just happened that this stand got marked a little heavier, and we really wish that this difference was a little greater. As you can see, we have about 13 square feet of basal area difference in these 2 treatments, and so the gradient's not quite as great as we would have wished. So what did these stands kind of look like in terms of their species composition? I'm not going to show all of the treatments, I just wanted to give you an idea. So what you're looking at on the left is a pie chart that's breaking down the pre-treatment basal area per acre as a % of the total by species. And I tried to color code these a little bit. And you can see we had a good amount of oak out there in the overstory, a little bit of ash, some poplar, a good amount of sugar maple. Hickory is our next largest percentage after our oaks, thus the oak hickory categorization, and then a whole suite of others. And when we mark these stands and harvest them, remember I said we retained oak and ash in particular, and then persimmon, which would fall in the other. And you can see we've made a huge difference on the species composition of the oak. We left oak preferentially out there. Ash really didn't change that much. We took out from the poplar, the sugar maple, and the hickory component and the others. That's where we were harvesting from. In contrast, the same pie graphs with the same idea, except now this is for the 75% retention. And I hope when you look at these 2 pies, what goes through your head is that they look awfully similar. Because again, the goal of this was to reduce the mid-story and to not make gaps in the canopy. And we really didn't have much of a change in the species composition here. And that was not a goal, so we were glad with this response. So how did these treatments look? I'm going to show a couple graphs that look like this. The treatments along the bottom, the 0 being 100% being the control, the 0 being the clear cut. We've just lumped all the hickory species together for all of this analysis, and this is the basal area. The light blue bar is the basal area of all the species prior to any treatment. The dark blue bar is post-treatment, and for this analysis I did 5 years post-treatment. And then the kind of the darker orange colored bar is the hickory basal area before we treated the stands, and then the lighter is the hickory after. And as you can see, the means that have the same letter are not significantly different. So we didn't have any differences pre-treatment. They all were very similar. But post-treatment, we ended up with 3 very distinct basal areas. The control and the 75% had significantly more basal area than the 50 and 25, which had more than the clear cut, and that trend held true for just looking at the hickories as well. Same graphs, the same type of schematics here, except now we're looking at trees per acre. And you can see we again have the same type of gradient that we created. And so we have more trees out there in the control in the 75%, an intermediate level of trees per acre in the 50 and 25, and then obviously significantly less number of trees per acre in the clear cut. So one of the things that I've tried to emphasize is that we were trying to alter the light, because we believe this is the mechanisms that's driving some of the responses that we're seeing to the regeneration. So this little table is telling us a little bit about those light levels. So again, the same type of depiction for the different treatments, the control and the clear cut. And the middle column is the canopy cover, and this was done 1 year post-treatment. And we used a handheld spherical densitometer at these different measurement plots. We took several measurements and averaged them for this canopy cover, and I just picked 1 month that we happened to use here. As you can see, there was no difference in canopy cover in the control and the herbicide treatment. Again, we were trying not to make holes in this canopy. We had almost the identical amount of canopy cover in the 50 and the 25, and then we had a significantly lower amount of canopy cover in the clear cut. And if anybody's wondering how we had 32% of canopy cover in a clear cut 1 year post-treatment, I would welcome you to come to Alabama after we do our next study in which we clear cut and see this tremendous amount of growth that we have on these good sites. So this is very much a legitimate number for our clear cuts. Then the last column, we were looking at the percent of full sunlight measured at breast height at 4.5 feet. And how we do this is we take one of these septometer devices and we put it out in the open, and we take another one and we walk transects through all of these stands, and we just take hundreds of measurements. And then we compare how much light we're getting underneath the canopies compared to what it would be out in full sunlight. So full sunlight, you would be 100%. So what percentage of full sun are we seeing in the control? And you can see we're getting about 8%. And it's pretty dark under there. If you move down the chart here and you look at the herbicide, even though the canopy cover was the same, we had twice as much light that was getting in this herbicide treatment compared to the control. Interestingly, we had twice as much from the herbicide, twice as much light in the 50 and the 25, and then twice as much that was hitting the forest floor in this clear cut. So we have one gradient of canopy cover that then also lent itself to having a gradient of light as well. And this is a picture of these devices. This is the handheld spherical densiometer, and this is the septometer. This is the small computer, and this is the wand that has the photoreceptive cells on it. And this is just looking for the herbicide treatment. I think it's interesting to look at it. So we didn't have light treatments, pre-treatment, but this is in August. These are the measurements we took over time, and this is just a percentage, and you're looking at the canopy cover or the percent full sunlight. So there you can see a pretty nice pattern here, which is we didn't make much of a difference in the canopy cover, but after we treated it, we had quite an increase. If you can remember, the control was down here about 8%, which is where this would have been. So we had an increase, but it was somewhat ephemeral. This burst of light that we saw only lasted for a couple years, and it has dropped down and it's remained fairly low in this treatment. If we contrast that to one of the clear cuts or the 0% retention stands, again, we're looking at the same graph again. You can see that after we harvested, we definitely had an impact on canopy cover, but over time that canopy cover is increasing. We just have a lot of trees that are regenerating and coming back in this stand. And conversely, the amount of light, we had a big burst of light a couple years after harvesting, and now the amount of light that's reaching that forest floor is starting to decline. And finally, if we look at, I just picked the 25% retention stand, we have, again, a similar type of pattern. You can see after we harvested, the amount of canopy cover dropped, and it's now increasing as those retention trees are taking up that space. And then our light kind of really peaked 2 years after, and then it's dropped down, and interestingly, we're actually getting a lot more light out there right now than we were before. And we think that this increase in light, a little bit of an ephemeral effect here and then an increase, probably has to do with reaching a stem exclusion stage. We had stand initiation in the understory here, all that regeneration taking up this growing space and taking up this light, packed in there pretty densely, and now some of those stems are starting to die, and that's allowing a little bit more light in. That's what we think is happening there. So kind of a busy graph, but interesting to just look at the absolute values. So these are the seedling counts per acre. This is all natural regeneration, and this is for the herbicide treatment, the 75% retention. And I'm presenting two numbers, the pre-treatment numbers and the post-treatment numbers. And these are all the number of seedlings per acre, so an actual tally. And then what I did was I broke them down by seedling height classes. So these are the small ones, less than 1 foot. These are the medium size, 1 to 4.5 feet, which we know would be much more competitive. These are our winners, the 4.5 feet ones that are greater than 4.5 feet, and then here's just a total. So if we look at pre-treatment and post-treatment, we can see a slight difference in the total number of seedlings, definitely seeing these small seedlings dropping, and some of this is just natural stand dynamics, but some of this we believe, if you see there's almost a 2,000 seedling per acre increase in this medium-sized class, and we have some of these small seedlings that have now grown up into this size class in particular, and that's when we look at all of the seedlings together. If we look at the topic of today, which happens to be hickory, remember this was the oak shelter wood, but we're curious as to what hickory did. We see very little difference in the total number of seedlings. It went from 384 to 348. Again, some natural stand dynamics going on there. We had hardly any difference in the small, absolutely no change in the medium, and very, very small change in the large. So the hickory at this point in time is not responding to this treatment at all. If we contrast that with the oak, which is the next one out here, you can see we had a lot of oak out there. We had 1,500, which is a lot for us before treatment, most of them in this small size class. We have a significantly less number of oak out there now. But what we're kind of excited about is, even though we have dropped the number of small seedlings, we've almost doubled the number of medium-sized seedlings. We didn't have any large oak reproduction out there, and now we have 9 seedlings per acre that are 4.5 feet or taller. And so the question is, if you take your 55 and your 9, is that enough oak per acre? How many of those will make it through the removal of the overwood, and is that enough to sustain the biodiversity in your stand? And that's one of the things we hope to follow. Contrasting to that, what really responded in these stands were the sugar maple, and you can see the overall totals didn't differ too much. We lost these little ones, and we had a tremendous number of sugar maple out there that were in this medium-sized class, and when we reduced that mid-story, they quickly grew from this into this size class, and we also had it going this way as well. And you can see we didn't have any big ones, and now we have at least 6 per acre. And this is in contrast to David Loftus' research, and because he didn't have sugar maple. And again, remember the impetus of this treatment was to alter the light so the small advanced reproduction could grow in a more competitive position without allowing yellow poplar in. I'm not presenting yellow poplar today, but we did also allow some yellow poplar into this stand, but we also enhanced the sugar maple out there. So again, a goal was to evaluate the response under these light levels, and we wanted to reduce the mid-story to grow this oak, and the main competitor was supposed to be this shade-intolerant yellow poplar, which we certainly have as one of our competitors. This is a picture of one of the work-study students out on one of these stands. And this is this post-treatment sugar maple layer that really took off after we reduced the mid-story. And it's just an interesting contrast to what David found on a little bit different site. So one more seedling count. This is for the 25% retention. As you saw, we let a lot more light in here. There was a lot more disturbance going on. And I just want to talk about the hickory in particular. And you can see we had 478 hickory pre-treatment, 417, again, some natural dynamics here. We certainly lost some of these smaller ones during the harvesting activities, but you can see we've had this tremendous increase in this middle size of hickory. So right now we are pretty confident that the hickory are responding to this huge increase in light. But just quickly as a side note, if you look at the medium-sized sugar maple, they also are responding to all this light. And if you look at this medium-sized oak, they too are responding. Everything has responded at this point. We're not quite to the point yet where we can decide who's going to win, but everybody certainly responded. We tagged, as I mentioned, a good number of seedlings out there, and so I pulled out just the hickories, and I looked at their survival percentages, and we had no significant differences among treatments by year were found for survival. So the treatments are not having any effect on actual hickory survival, but they are having some effect on the growth of some of this hickory. So what we're looking at in the top is the relative height growth in centimeters over time, and on the bottom is the relative diameter growth in millimeters over time. The orange is the 25% retention, the light aqua color is the 75, the yellow is the 50, the black is the control, and the blue are the clear-cut seedlings. And you can see in 2006, after 5 growing seasons, the relative height and diameter of the hickory seedlings in the control, which are the black, were significantly less than the growth of the hickories in the 25%. So here's the control, and here's the 25%. And if we look at the basal diameter increase, we also had a similar trend. The control seedlings had about 4 centimeters of relative height growth over 5 years, versus the 25% retention, which had 48 centimeters of height growth. And I also want to point out, if you look at the top three here, you see that the ones that had the most disturbance and let the most light in were where the hickories responded the most. And finally, one of the other things I mentioned was we're really interested in sprouting probabilities because we know that these sprouts from stumps that were cut contribute to the regeneration of the next stand. So what I did was I took the diameter of the hickories in the 3 cut treatments from 0 to 25, and I looked at their probability of sprouting or not. So this was just for the 3 that were cut. And you can see if you look at about a 10-inch diameter hickory has about a 45% probability of sprouting. Of course, these smaller hickories have a much greater probability of sprouting, and like many hardwoods, as you get a larger diameter stump, the probability of sprouting decreases. So with that, how can we regenerate hickory on good sites on the Cumberland Plateau? Well, if we compare what we might predict the type of species composition that we could have with our regeneration goals, and we're doing this using David Loftus' regen model that he's talked to you about, and we're calibrating this for the Cumberland Plateau. So we used his model to predict what these stands would look like if we did a regeneration harvest. We know that oaks are going to lose out to yellow poplar, and in our case, they may also lose out to sugar maple. And really, hickory is a non-player. It's kind of maintained itself in the same numbers that it had regardless of treatment. I think we need to really think a little more about how much we can rely on these stump sprouts. As I told you, 20% of these stands are in hickory. I showed you the sprouting probabilities, and for a 10-inch tree, there was approximately a 50% chance of that hickory sprouting. And there were on average about 20 hickories per acre in this 1 to 10-inch DBH class. So if you had 20 hickories in this DBH class, and those were going to get cut, half of those are going to sprout, and we need to follow those over time to see, of those sprouters, which ones are going to contribute to the next stand. And we also need to add some other variables that I've talked about collecting, that we've collected: slope, aspect, elevation, and of course all this light data that we have. Finally, at the stand level in the heavy cut stands, this is a 50-25 in the clear cut, hickory regeneration in the 1 to 4.5 foot size class increased, but so did everything else. So this large pulse of light that we when we opened up these stands really caused everything to respond, and we need to continue to follow this over time. We're in the middle of it, and definitely get the overwood off so we can see if the shelter wood really did make any difference. Just kind of a take home, we chose very high vigor leaf trees. The landowner was gracious and allowed us to leave some big money trees out there. And we didn't have any blowdowns. We had one tree that died in the control, and we had very negligible epicormic branching. So we chose high vigor leaf trees, and that's paid off, and that those trees, although it was a risk to leave them out there, they have maintained themselves, and they will be available for the next harvest. We're starting to question if we need some type of a combination treatment. When we reduced the mid-story, we had the sugar maple take off. We also had poplar take off. We had oak take off. We observed out there that where they took off the most was where there was a little bit more light. Remember, I told you how ephemeral that light was, especially in the herbicide treatment. So do we need to do this mid-story reduction, open up that growing space, but knock a few holes in the overstory, where on the peripheral of that big light patch, we may be able to better regenerate our oak. Of course, in the middle, the yellow poplar is going to take off. I'm not sure how this would be accomplished, and I'm not sure if it's feasible to do it, but it's certainly a way to look at trying to get to the regeneration goals that we have. And one of the things that we've come away with is that our knowledge base is much greater for oak than it is for hickory. And when someone asks about how does hickory regenerate, or how do we regenerate hickory, I think it really is an excellent conservation question, especially in today's environment, where we know that all of these species are important, and that we really have a goal to maintain these species in our stand. And we have to hedge our bets, not only for timber, but for other resource values out there, such as wildlife, aesthetics, and so forth. And so it's been an interesting conservation question, and I think we need to continue to follow the hickory and hopefully do a little more work with it. A lot of people have participated in this study. There's been a good number of students from Alabama A&M University who've assisted. The study was originally put in on Mead Corporation property. They had provided me a lot of assistance. But I want to recognize in particular Greg Jansen, who's with Stevenson Land Company. He was the lands manager with Mead, and now is the lands manager with Stevenson Land Company, and he's helped drive this study. And Ryan Sisk is my lead technician, and he is the person that has been very diligent about all of the data collection out there, and I couldn't have done it without those two guys. With that, I thank you for your attention.